Great. Yeah, sorry I can't be here in person, but I really wanted the chance to maybe talk about a bit of the DSci-ish or proto-DSci work that we've been doing, and it's fun to think about where this might go in the future. So I run a group at MIT that works on technologies for mapping and controlling the brain. And recently, we've also started to think about how to apply these tools towards modeling the brain. Now, for me, the core motivation is really philosophical. I really, ever since I was about 9 years old, have been just sheerly puzzled by human existence, and wondered, what are we? Why do we do what we do, think what we think, feel what we feel? Why do some of the objects feel things like us, and others, like trees and rocks, presumably do not? Um, and how can we be happy? How can we achieve meaning? Is there such a thing as a new kind of enlightenment that we could perhaps invent if we understood more about how we existed? So that was my childhood dream, and it was a long journey to train as a physicist and then as an engineer. And then as an adult, I entered brain science. And it also became clear that brain science was full of lots of other pressing needs. Over 1 billion people around the world have some kind of brain condition, Alzheimer's disease, ALS, spinal cord injury. The list goes on and on. None of these can be fully cured, and the treatments tend to be quite partial and have lots of side effects. In fact, these are some numbers from 2010. I think they've actually gotten worse. But you know, to take a therapy out of the lab and into the marketplace could take a decade. The failure rate is very close to 100%. The cost is enormous, perhaps over $1 billion each in US dollars. And even if you make it all the way through those hurdles, the success is often quite partial. The treatments often don't work very well, and the side effects can be quite prominent. So, what can we do? Well, I thought a lot of the history of science, and I trained as a physicist. And one very intriguing point, I think, that you can see from the history of science is that when a scientific field hits its ground truth, and one way to think of that might be what are the fundamental elements, the building blocks, if you will, and how they interact, what emerges from the building blocks that make up a system. Then you can get to a period of great progress. Maybe the most famous example is physics, where quantum mechanics yielded the transistor, which of course, led to the microchip, the laser, of course, um, the internet, wireless telecommunications. The list goes on and on. So physics is a great example where really understanding the building blocks and how they work together was able to yield a huge amount of progress that was not only foundational, but augmented everyday life. Chemistry, you could argue, also went through a similar renaissance with the periodic table and the molecular bond. What about the brain? What's the ground truth for the brain? Well, biology is different from physics and chemistry, and at least a couple of regards. So, first is spatial complexity. The brain is a large object, right? But the cells that make up the brain are full of nanoscale tendrils and connections, and the geometry of those tendrils and connections is extremely complicated. A cubic millimeter of the brain might have 100,000 brain cells with 1 billion connections, like the one we just zoomed into, called synapses. And the synapses and the cells are full of molecules, biomolecules. The human genome has about 20,000 or so genes. Those genes encode for countless kinds of biomolecules. So, how can you understand a system across all those spatial scales? But it's worse. Time is also an issue. So, if you are interested in Alzheimer's disease or learning or development or aging, those are slow processes that can take years, even decades. But the fastest events in the brain are quite brief, biologically speaking. Millisecond timescale electrical pulses within brain cells and millisecond timescale chemical exchanges between brain cells. So I sometimes only half jokingly say that our problem is simple. We just have to conquer space and time. Okay, so to summarize the problem, we need a couple technologies, right? Ideally, those high-speed electrical signals, we could control them to figure out what they do and observe them as well. Those molecules, those wires, those synapses, we should ideally be able to map all of them. And such data sets, of course, are going to be extremely complicated. If we want to understand how these building blocks of life work together to mediate brain computations, we ideally need to simulate how they work together in computer software. So technologies for recording and controlling high-speed signals could be used at the same time, and then mapping technologies could be done and applied to the brain after the recording and controlling is complete, and such data sets could then be integrated. So computer simulation of the brain could potentially satisfy some of those goals that we outlined in the first slide. Maybe we could pinpoint therapeutic targets in the brain and help address conditions like aging or specific diseases. Maybe we could explore how brain cells interact to create thoughts and emotions, and maybe even touch upon the philosophical mystery that is consciousness. And of course, all too obvious in this day and age, but AI is of great excitement. If you can inspire new, more human like AI with human like creativity and even ethics, that could be quite interesting as well. So I'll tell you a couple of short stories about technologies we're developing, as well as sort of the DeSci prototypes that we've been thinking about over the years. And it'd be fun to think about where this all might head. Let's start with controlling the brain. So brain cells compute using electricity. Of course, you can't just apply electricity easily and hope to hit a single cell, right? Because electricity would go in all directions. If, though, you could install a little solar panel on a brain cell and then shine light on that brain cell, the solar panel would convert light to electricity, and you would be able to activate or shut down the electrical activity of that brain cell. You could dial in a therapeutic recipe, even perhaps someday. And the brain doesn't have pain sensors. You could bring an optical fiber into the brain the same way that electrodes are brought into the brains of even awake human patients undergoing deep brain stimulator implants. How to make brain cells sense light? Well, it turns out all over the tree of life, you can find molecules that convert light into electrical signals. And so in this animation, this is a single-celled green algae, and it has a little eye spot, that brownish spot in the back, which senses light from the sun and uses the sunlight to control a series of signals to control flagellae or tails to help the algae swim around. We just zoomed into the eye spot. The eye spot is full of light-activated proteins that when shined light upon, they will open up a pore and let charged particles or ions across the membrane of that eye spot. Then, through a series of signals, you get control of those flagellae or tails so the algae can swim and be near the surface of a body of water. So, this is where we got very lucky. People kindly shared the gene that encoded this protein with us. People have been studying these kinds of proteins for many years, over half a century, I guess, at this point, for biophysical and bioenergetic investigations. And we obtained the gene that encoded for this light activated protein. And this is where serendipity hit. The gene's small enough that we can deliver it to a brain cell using a small virus. The brain cell took up the gene and was able to manufacture the light activated protein. That was serendipitous, right? It didn't have to work. And then maybe even more serendipitously, when we shine light on the brain cell, we were able to drive the brain cells with electrical pulses, not unlike the electrical pulses that are in your brain right now as you hear me say these words. So that was a lot of luck. The molecule could have been toxic, maybe it would not have worked, but it did. Okay, this next slide has a movie with flashing images. So if somebody is photosensitive, maybe don't look at this until I get back to you. So here is an animation explaining how we can use this technology. So cells in the brain, this is a cartoon, are firing off electrical pulses. We can deliver the gene that encodes for the light activated protein, or rhodopsin, to just some cells, for example, and not others if we so choose. So the cells that just lit up in green are little star-shaped cells called basket cells, a kind of interneuron. And if you drive the brain with light now, the cells, the basket cells that have this light activated protein will respond, but their neighbors will not respond to light. Okay, the flashing images are now over. Okay, so this is where I would love to talk a bit about sort of the DeSci journey that we started upon. We invented a tool set like optogenetics, as we call it, opto for light, and genetics because it's a genetically encoded tool. And it's inexpensive, and we can disseminate it very, very effectively. And it gives you this sort of ground truth neural code. We deploy it. We share it as freely as possible for people to use to make discoveries. And then finally, we design. We can come back into the game and help people craft, based upon their discoveries, practical applications that benefit human well-being. I should highlight a nonprofit called AdGene was really key to making this field what it is. They disseminated the DNA encoding for these optogenetic molecules from our group, and now they disseminate, who knows how many molecules from thousands of groups around the world. And so they've sent these to tens of thousands of destinations over the years. Okay, so what kind of invent, deploy, design, as I like to call it, examples come to mind? Well, I love this example from Li Wei Sai's group at MIT. She studies Alzheimer's disease and drove brain cells in mice that model Alzheimer's disease at a specific rhythm, 40 times a second. If you do that, strangely enough, the brain starts to clean up the molecular hallmarks of Alzheimer's disease. You clean up plaques, you reduce inflammation, you improve blood flow. Now, how do we make a practical version of this? Well, the teams under Li Wei's leadership went on to design flickering lights and clicking sounds, basically movies that could replicate this brainwave, this 40 hertz gamma oscillation. And in animals, that was able also to clean up the molecular debris in Alzheimer's and to improve cognition. And so now we actually spun out a company and are running a large scale clinical trial to try to see if we can help confront Alzheimer's disease with basically a movie. That's a great example, I think, of invent, deploy, design, and action. I'll give you one more example: blindness. Millions of people have photoreceptor loss forms of blindness. This is a mouse model of blindness that recapitulates such a condition. The photoreceptors are atrophied. The mouse is in a water maze. So mice love swimming, but they love sitting and doing nothing as well. And so the water maze has 6 arms, and the mice swim around. And if they find a platform under a lamp, they can crawl out like that mouse just did. But these are mice that are blind, and so they don't see the platform. It takes them a while to find the target. Now here's a mouse that was blind a few weeks beforehand, and now can go right to the target. We treated it with 1 dose of a gene therapy vector with our light activated molecule, basically turning the spared cells of the eye into a camera. We share these tools with many people, and a team in Europe was able to take one of our molecules and, with a blind person, help restore some degree of functional restoration. He could recognize household objects. He could see doors in a hallway or lines of a crosswalk. Not perfect vision. He was not able to read fine text or recognize faces, but still very exciting to see the beginnings of a new arena of therapy. So those are 2 examples where we invented a tool, we shared it with people who then made interesting and powerful discoveries using it, and then we helped them design approaches that are practical to help people that are now in human trials. In the interest of time, I'll just tell you about one more story, which is both important for brain simulation, but also fits this model: mapping. We want a map of the brain, how our brain cells organize into networks, how our molecules organize within brain cells. I think we've all seen pictures like this, brain scans. They use MRI or other methods to image the brain non-invasively. And non-invasive is powerful, but you're not able to see individual brain cells or connections, which are much, much smaller. Each blob or voxel might contain millions and millions of brain cells. At the other extreme are microscopes. They let you see tiny things, but the wiring of the brain and the molecules that make up brain cells are nanoscale. You can't see them with an ordinary microscope. We had the idea, what if we just took the brain and made it bigger? The material in baby diapers is a swellable material. You can add water, and in this cartoon, as you can see, it'll absorb the water. The polymeric threads of the baby diaper material, shown as white lines, will spread apart from each other, and you'll get a swelling. We wondered, what if we could install those polymeric chains of baby diaper material inside the brain? If we chemically could weave a dense spider web-like mesh of baby diaper polymer, sodium polyacrylate for the aficionados, in between cells and around cells, inside cells, in between biomolecules, around biomolecules, do it just right, add water, and the polymer would swell and pull all those biomolecules, the building blocks of life, apart from each other. So this will not work on a living thing, of course, but you could use this to get the molecular anatomy of an object. And we found that it would work. Here in panel B is a small piece of the mouse brain, a couple of millimeters on the side. Panel C, about a day and a half later, we've swollen it 100 times in volume, about 4.5 times in each direction. And the polymeric threads start out very dense, like in the cartoon in the upper left, and then end up like something in the lower left. Here's a time-lapse movie of half an hour sped up to half a minute, where we formed the polymer throughout the brain earlier, and then we added water right there. And I hope you can see this piece of brain tissue is growing before your eyes. Okay, that's what it looks like to the eye. What's happening at the nanoscale? Well, what we're trying to do is to take a brain cell like this golden neuron on the left and make it look like something on the right, a constellation of biomolecules hovering in space. They've been pulled apart from each other, and that way we can image them and resolve them from each other using an ordinary microscope. What's happening chemically? This is a bit of a technical aside, but bear with me for a second because I think the payoff is worth it. In this cartoon, I'll explain to you how it works. What's happening at the nanoscale? So in this cartoon, proteins and other biomolecules are shown in brown. We had to invent anchoring molecules that would bind to these proteins and other biomolecules so we could pull them apart. Think of it as attaching little handles to everything so that we can yank on them and pull them apart from each other. Then we had to weave this dense spider web-like mesh of baby diaper polymer. We had monomers, little white spheres in this cartoon, and they self-assemble through a process called polymerization into these polymer chains that weave their way inside and outside cells and in between and around the biomolecules. And importantly, when those chains encounter a handle, they form a bond. We're almost there, right? We have a way to swell the brain and pull the parts apart, correct? Well, almost. There's one more step we had to figure out, which was softening. The molecules are very happy where they are. We have to loosen them up and get them to break their allegiance to each other using chemicals. And then we add water, and because of the softening, and because of the polymer, and because of those handles, we can now pull the building blocks of life, biomolecules, away from each other. We can then label those biomolecules with all sorts of different labels, like fluorescent or glowing molecules. Here's an actual piece of brain tissue from the hippocampus of a mouse. It's part of the brain involved with memory formation, amongst other things. And the brain cells are expressing fluorescent proteins, so they glow different colors. And we expanded it. And I hope you can see now that we can see the fine tendrils of wiring of the brain. Now, a big focus of our work, and I think this is very amenable to the DeSci philosophy, is to make things really easy to use, right? Optogenetics, which I told you earlier, is being used by thousands of research groups. Expansion microscopy is being used by thousands of groups. One of my favorite quotes from this recent news article about the technology, it's so easy, even my 9-year-old can do it, said Dongshin Zhao at Carnegie Mellon. Okay, so let's get back to this idea of invent, deploy, design. We created a tool that can allow people to probe the ground truth of the brain. We deployed it, just like optogenetics is being used by thousands of people, expansion microscopy is being used by thousands of people. But here's something interesting. Expansion microscopy is not only easy to use, it's easy to modify. And so I'll share with you one last sort of scientific story before getting to my conclusion, which is a group in Germany led by Silvio Rizzoli, was able to modify the technology and make it apply to a different kind of problem. So again, we invented the tool, we deployed it, and then working with others, we could help them design variants and adaptations of it. And here's what they did. Instead of pulling molecules apart from each other, what they did was take a molecule, anchor it to the polymer, chop the molecule into pieces, that's the new part, and pull the pieces apart from each other. And if you do that, you can kind of see the shape of the molecule. This is pretty cool, right? In gray is the ground truth, the known structure of molecules through classical techniques, X-ray crystallography, cryo-EM, now AlphaFold prediction. The false color images are single unaveraged expansions of these molecules. So it's fun to think that now with this variant that they developed on expansion, that you can visualize the shapes of molecules with ordinary confocal microscopes, in this case with the SURF super resolution configuration. Now, what we want to do now is to integrate all the different technologies towards modeling the brain. Can we record and perturb neural activity, as shown in the upper left of this cartoon, and then build molecular maps, as shown in the lower left? We can do that through optogenetic means and expansion means. Then we can make models based upon top-down machine learning inference. We could also do inference of underlying physiology of the models. And then finally, from the molecular maps, we could try to do bottom-up models. And we're doing this with a large and growing cast of characters, including many of the people named at the bottom of the slide. Our hope is to start with small brains, but to scale as rapidly as possible. Worms and fish have a fairly small number of neurons. Mice are bigger, and humans have much bigger brains. And so a very fun question then is, what versions of invent, deploy, design might we be able to encounter and explore in the time to come? I think of, in these short form talks, it's difficult to acknowledge all the people who contributed to the work, but I've had their names at the bottom of the slides. And here's an even longer list of people who have helped with these and other projects over the years. Our website is synthneuro.org, and my email is edboyden@mit.edu if you have questions. And I'd be happy to take questions now.
Thank you so much, Ed. That was extremely riveting. I'm just so grateful that you're trying to push this forefront forward on multiple angles. We have just a few minutes for Q&A, so I'm going to lead the discussion and then pass it off to the audience for questions. The first question that comes to mind here is, why do you think, or maybe I can frame this a little bit differently, there's so much promising science happening here. Why don't you think the science has progressed farther or faster than it has? What are the bottlenecks holding back your research from truly accelerating 10 times faster than it currently is?
Yeah, it's a great question. I think two things come up over and over again. I mean, I think this sort of bottom up way of thinking about science, right? Of really understanding all the building blocks and how they work together, um, is somewhat different than sort of a classical hypothesis driven way of approaching science, right? But as a physicist who just wants to see the parts and how they work together, it seems very natural to me. So it's always been a bit of an uphill battle to, um, raise resources. In fact, when I was looking for a faculty job, I even to set up my group and to do research. The majority of the places that I applied to actually turned down my application at the time. I was very lucky to find a home base in a interdisciplinary department at MIT, where I then was able to develop a lot of these approaches and ideas. Um, the other thing that is sort of very interesting, and I tried to highlight this in the talk, is also just the extraordinary, almost omnidisciplinary need for knowledge. And this is why this sort of DeSci approach in philosophy is so appealing, and why we've tried to follow, um, such kinds of principles from the very beginning. You know, for optogenetics or expansion microscopy, we needed genes, light, hardware, chemistry. It's not just interdisciplinary science, it's omnidisciplinary. And so, how can you bring people together or facilitate people to work together across every boundary possible? And that's why, with the three stories that I highlighted, the Alzheimer's therapy in trials, the blindness treatment in trials, and this version of expansion to see the shapes of molecules, I tried to really highlight how the need for expertise from sometimes very, very different sources and directions is not just helpful, but perhaps essential.
Definitely. I think it would come as a surprise to many here. It came as a surprise to me when I first talked to you. When you explained sort of the challenges or the lack of excitement when you first tried to describe the early research that you were doing, such as expansion microscopy and optogenetics. Could you share a little bit about that story for folks here to learn about what your struggles were like trying to get your research initially funded way back in the day?
Sure, yeah. I only recently started publicly talking about this. I gave my high school graduation speech back in Denton, Texas, a couple of weeks ago, and then I actually, for the first time, publicly talked about how the vast majority of places I applied to for faculty jobs turned me down, and it was hard to, yeah, it was a bit surprising that people didn't really like, for the most part, the kinds of things I wanted to do. Um, yeah, it was sheer luck that helped me get a job. I was a teaching assistant for a professor. And I worked as an undergrad in the lab of another professor, and they worked in this interdisciplinary department at MIT where they just happened to have a job search that I guess they didn't find a candidate that they liked. And so that's how I got that faculty job. In 2015, when we came out with expansion microscopy, we kind of had a similar problem. We had a lot of struggles to get government grants. I think we had like 9 government grants rejected on the topic. Um, and it was sort of sheer dumb luck. That seems to be a common theme of what keeps our group going. That resulted in us getting funded. So a member of our group was friends with a grad student who, it's a long chain of personal connections to the foundation known as Open Philanthropy. And they, at the time, I don't think really funded much science, but they offered to investigate what our group did and try to understand how we did things. And you can read the full report on their website, but it's like a 30-page report or something. But in the end, they said, "Wow, this seems like something that we should support." And so that's what kept us in the game. So it's always a lot of serendipity to keep things going. And I guess one reason why I'm interested in thinking more deliberately about DeSci and such themes in the current day is, can we do on purpose what in the past we did accidentally, right? So I gave a couple examples where invent, deploy, design kind of worked through, unstructured luck. But can we be lucky more on purpose here?
Yeah. Yeah, and that's definitely something we've been focused on, how to actually engineer these kind of outcomes instead of hoping that through luck or through serendipity, these kinds of things can naturally emerge. I'm wondering, that there's an abundance of need for scientific funding right now, especially with the day and age that we are in currently. If you could imagine that we could expand scientific funding by 1 or 2 orders of magnitude, what effect do you predict that would happen for society and for humanity globally?
Yeah, I think now is an extremely interesting time where science funding could galvanize um, real revolutions at an accelerated pace. And the reason is, I think that technologies are getting to the point where you could really try to describe a complex system in terms of all of its parts. I entered neuroscience about 27 years ago when I did my first undergrad research in neuroscience, switching from physics. And in those 27 years, I don't think we've cured a single brain condition. And even the treatments are partial and have a lot of side effects. So it's very clear that I think the big revolutions in terms of neural therapeutics are in the future. And so, but as I hope I've tried to persuade you over the last 25 minutes or so, the tools that we've built for mapping, for controlling, they're getting to be really good. And so, my excitement and dream is that in the coming years, would it be possible to map out what an aged state really is, to map out what a disease state really is, and to pinpoint exactly where to intervene in order to resculpt that system towards a healthy or more well-being oriented state. um, as a very sort of bottom-up thinker who wants to see the parts and how they work together, um, it seems like that dream might be coming into view. So I do think that the coming years are going to be tremendously exciting. But back to your original point, I think funding to drive that forward will be, of course, essential.
I think it's something that we often say in the crypto space, which is it was easier to invent Bitcoin than it was to reform the US government or the Federal Reserve. And similarly, we think it's easier to create an alternative to traditional scientific funding than it is to try and reform the currently existing system. If you were in charge of building this new system, how would you like to see it operate? How would you engineer? What kind of incentives would you put together to have a system like this optimized for impact?
Mhm. Well, a couple thoughts. One thought is, how do we evaluate scientific plans? I mean, it's very hard to do that, right? Um, but I do think one could try to build methodologies of evaluation that are based upon first principles thinking. If somebody's going to propose a new chemical tool or physical tool to investigate a biological situation or to confront a biological disease state, well, let's analyze it in terms of the underlying physical and chemical principles, right? Are there ways to evaluate science that are more structured and based upon, maybe deconstructive and analytical techniques that could be systematically applied? The second thing is serendipity. I mean, if you look at the history of science, look at CRISPR, look at fluorescent proteins, look at all the different, next gen sequencing, the different technologies that have revolutionized biology medicine. They came from different people. They came from all over the globe. They came from collisions of fields. People often talk about, like, Bell Labs, right, which had all these storied innovations in the 20th century. Some of the most important programming languages. The precursor to the laser, the discovery of the cosmic microwave background radiation, the transistor, of course. And a lot of people then try to replicate, Bell Labs, right? But if you look at empirically where innovation pops up, the Bell Labs of the future is the whole Earth, right? And so, is there a way to find projects and people as early as possible, and then to help nurture them? And then the final thing that I'm excited about is connecting application and origins. So by the time something is commercially viable, um, maybe the precursor inventions, the precursor discoveries are so far back, and that leads to two kind of incentive issues. The first, of course, is that the people who developed the initial discovery or invention, they were so far ahead of their time, it was hard for them to get going. And then finally, the final commercial application usually is so far downstream that the originators aren't necessarily rewarded for their efforts. And so I'm very intrigued by blockchain-like or other ways of connecting origins of ideas to outcomes, both to reward originators of ideas and the beginnings of things, but also to connect resources to such people so that they can get going.
No, I absolutely agree with you. And from what we've seen, talent can come from everywhere globally. And we're building a new system to allow anyone anywhere in the world that wants to be a scientist actually have access to this ecosystem. Thank you so much, Ed. And let's give a round of applause for the legendary Ed Boyden.
Yeah, please stay in touch. We'd love to have more conversations. And yeah, sorry I can't be there in person.